My name is Étienne, and today I'll talk to you about my project, which is to build a machine learning-based workflow to predict snoRNA abundance status across species. So first off, snoRNAs are small nuclear RNAs, so non-coding RNAs that can be separated in two types based on their structure and function. As you can see, both snoRNA types have their respective box or motifs displayed here. In human, most snoRNAs are encoded within the introns of genes called host genes, and a great proportion of snoRNAs are also encoded within intergenic regions, depending this time on their independent promoter to be expressed. There are several functions associated to snoRNAs. Their main function is to guide RNA modifications, such as the 2' O methylation for CD-box snoRNAs and the pseudo relation for HCA-box snoRNAs. This can be done through the complex they form with different core proteins and enzymes called the snoRNP. Their canonical target is ribosomal RNA, but they can also bind different other RNA types, such as pre-mRNAs and transfer RNAs. And the modification they guide on these RNAs are really important in, for example, ribosome biogenesis, but also in splicing regulation, pre-mRNA stability, and polyadenylation regulation. We recently generated several RNA-seq datasets using a thermostable reverse transcriptase called TIGERT, which helps to reverse transcribe highly structured RNAs such as snoRNAs. So we generated these TIGERT-seq datasets in 7 healthy human tissues, as you can see here. And what we found in that study is that when we filtered snoRNAs based on their abundance, so greater than 1 transcript per million or TPM in at least one of these tissue, we found that only a third of all annotated human snoRNAs were expressed in these tissues. And of course, 2/3 of the other snoRNAs were not expressed in these tissues. What we found also in this project was that several host genes that contain multiple snoRNAs have snoRNAs that vary very much in abundance. So for example, a host gene that contains in its first intron a snoRNA that is highly expressed, and in another intron that contain a snoRNA that is not expressed or not detected in our data sets. So this study raised multiple interesting questions, such as why is such a small proportion of expressed snoRNAs in of all the annotated snoRNAs in the human genomes? And also, how can snoRNAs encoded within the same host gene vary so much in abundance? So these will be the 2 questions that I'll try to answer today for you. So I'll refer to, in the next few slides, to the abundance status of snoRNAs, which is basically expressed versus not expressed in our human tissue data sets. So in the literature, there's a few features known to influence snoRNA abundance, such as it was shown for a few CD-box snoRNAs that their distance to the branch point was critical for their expression. Was also shown for a few CD-box snoRNAs that the formation of a terminal stem was important for their expression. But our hypothesis is that there are many more abundance determinants or features that could influence the abundance of snoRNAs, because basically we don't know if these features apply to all CD-box snoRNAs, and we don't even have an idea if these apply to HCA-box snoRNAs. So the objectives of my projects are in 3 parts. The first one is to collect input features for all human snoRNAs. So we have both intrinsic features, so related directly to the snoRNAs, such as its global structure stability, its terminal stem stability, and also extrinsic features such as the position of the snoRNA within its host gene, related to the branch point, to surrounding exons, and also information on the host gene, such as the function of the host gene. So that's the first part. The second part was to train, using machine learning, to train the abundance status predictors. to predict finally whether snoRNAs were expressed or not expressed in our tissue data sets based on these features. So I did the hyperparameter tuning on a tuning set composed of 10% of these snoRNAs. Then I trained the models using a training set composed of 80% of these snoRNAs. And finally, I tested the performance of these models on an independent test set composed of 10% of snoRNAs. And if these models performed well, then I could interpret their decisions using SHAP values to see to give us insights about the main abundance determinants in human. First off, for the data collection, I'll show you here the distribution of terminal stem stability for CD-box and HAC-box snoRNAs. And as you can see, expressed CD-box snoRNAs tend to have a more stable terminal stem than their not expressed counterparts, and we don't see such trend in HAC-box snoRNAs. Another novel feature that I computed here was the box consensus score, which gives us information about whether the motifs within the snoRNAs are conserved or not. So basically, a score of 0 means that all of the boxes present in the snoRNA are identical to their known consensus sequence, and the more mutations the snoRNA accumulate, the higher the score is. So the less the boxes are conserved. As you can see, for both CD and HAC box snoRNAs, you can see that when they are expressed, they tend to have highly conserved motifs, which is significantly less the case for not expressed snoRNAs. Here I show you the distribution of global structure stability. We can see that for HAC box snoRNAs, when they're expressed, they tend to have a more stable structure than their not expressed counterparts, and we don't see that trend in CD-box snoRNAs. So here I showed you just a couple of distributions, just so we know there are 30 other features that can all be used equally by the models. So to know whether which feature is the most predictive, we'll just let the models tell us, basically. So for the second objective, which is the training of the model, we used 5 different predictor types, such as the logistic regression, the support vector, k-nearest neighbor, random forest, and gradient boosting. As I said in the beginning, we tuned the hyperparameters on 10% of snoRNAs, then trained the models on 80% of snoRNAs, and finally tested the performance on a remaining 10%. However, I'm interested in predicting the abundance status across all 1,541 snoRNAs, not only at 10% of that. So I did 10 different splits or iterations, as I call them, just to be able to predict the abundance status of each snoRNA once across these iterations. So here I show you the performance of the models. As you can see on this rock curve, all of the models across the iterations are highly performant. They have an area under the curve that is around 0.90, which is really great. And then I show you here the accuracy of these models, again, this time on the tuning, training, and test set. And as you can see, two types of models seem to overfit, as their training accuracy is very higher than their test set accuracy. So the gradient boosting and k-nearest neighbor, these two models were excluded for the rest of these analyses, and I kept only the logistic regression, support vector, and random forest for further analyses. Now for the third part, which is the interpretation of the model's predictions, since I showed you that the models are highly performant. As I said, we used Shapley additive explanations, or SHAP values. So basically, I computed a SHAP value for each feature and for each snoRNA in the test set. So here, for example, each dot would represent one snoRNA, We show here the distribution of these SHAP values for each snoRNAs per feature. So a negative value of a SHAP value represents the fact that this feature helps to predict, in our case, as not expressed. Conversely, a positive value of SHAP value represents the fact that this feature helps to predict the snoRNA as expressed in our data sets, and a SHAP value around 0 means that this feature has low impact on the prediction. So based on these kind of distributions, we can classify features by predictive rank. So basically, we'll first compute the absolute value of all of these values, and then compute the average of these distributions, which will give us something like this, these three averages. And based on these averages, we'll be able to define a rank, predictive rank. So the higher, the better. The most predictive feature in that case would be feature number 1, followed by number 3, and feature number 2, which would be the least predictive, so the less important in the prediction. So what I'll represent here is a graph with a lot of information on it. So I'll try to deconstruct it slowly. So first off, on the y-axis, you have that predictive rank that I just talked to you about on the last slide. So rank number 1 meaning that the feature is highly predictive, and rank number 30-ish is the least predictive feature. On the x-axis, you have all of the features that could be used by the models. These are ordered by the median of these distributions. So each distribution comprised 30 dots or 30 models. So as I said, we chose 3 types of models across 10 iterations. So that's why we have 30 models. As you can see on the top 4, we have in the first place the box consensus score, which is highly important in almost all models for the prediction. Then it's followed by the snoRNA global stability, and then the terminal stem stability, and finally the fact that the host gene is expressed or not expressed. Interestingly, if we looked at the distance to the branch point, which was known in the literature to to influence the abundance status of snoRNAs, we can see that it's really farther to the right, meaning that it's, according to our analyses, it's not really important. It's probably not the best abundance determinants across all of these possibilities. So on this slide, I'll try to answer one of the questions I asked in the beginning, which is why snoRNAs encoded within the same host gene can vary so much in terms of abundance. So here we have an interesting host gene called Cas5, which is a long non-coding RNA. It encodes 11 snoRNAs, 10 of which are expressed in orange, and 1 that is not expressed in blue here. So what's interesting is that our predictors accurately predicted the abundance status of all of these snoRNAs. So to represent this, we did a decision plot. So each line on this graph represents one snoRNA. Here the not expressed snoRNAs, and here the 10 lines for the 10 expressed snoRNAs. How to read this is from the bottom to the top. So we start from a base value, and then we follow the decision process of the model. So basically, either going to the left to the not expressed, or to the right to the expressed prediction. So what we first can see here is that most of these features here are not important in the prediction, since they do not help to move towards the right or the left so that much. And what we can see is that for the not expressed snoRNAs, it mostly has a lowly conserved motifs, and also an unstable global and a terminal stem stability, as it really helps to predict as not expressed towards the left here. Conversely, these expressed snoRNAs have highly conserved motifs and also highly stable terminal stems, which help them to be predicted as expressed snoRNAs. Finally, I applied the logistic regression predictor to various vertebrate species. First off, we processed tiger-seq data sets in mouse, and this is the results we obtained. So around 25% of these mouse snoRNAs are expressed in these conditions. Interestingly, and what's not shown here is that when I applied the logistic regression based on the top 4 features that I use, so box conservation, global stability, terminal stem stability, and whether the host gene is expressed or not, we found that almost exactly the same proportion, so around 25% of expressed snoRNAs. So here I represent you the predictions that we did on several other vertebrate species, ranging from the chimpanzee to the zebrafish. And as you can see, there is not much variation in the percentage except for the chicken. So most of these vertebrate species are predicted to have a low proportion of snoRNAs that are expressed in their genome. Interestingly, what we can see is that on top here we have the number of snoRNAs annotated in these genomes. And when we plot this data as a scatter plot, we can see that there is a nice anti-correlation between the number of snoRNAs per species and the proportion of expressed snoRNAs in that species, with the chicken and platypus being the two opposites here. To conclude, the working model we have right now in human follows as such. So we have expressed snoRNAs that are mostly intronic snoRNAs. 10% of these expressed snoRNAs are encoded within intergenic regions. Most of these expressed snoRNAs have highly conserved motifs and also a highly stable structure and also a long and stable terminal stem. All of these features help to potentially bind core proteins and enzymes to form a stable snoRNP, which can then act functionally in the cell, for example, to guide modification on their target RNA. Conversely, not expressed snoRNAs mostly are encoded within intergenic regions, and these snoRNAs mostly have poorly conserved box motifs, unstable structure, and also unstable terminal stems. We're not sure if these are actually transcribed since we don't detect them, but all of these features hinder their binding to core proteins and enzymes, and this results in unstable snoRNA, which is potentially rapidly degraded by exonucleases. So the take-home messages here that I have are that box conservation, snoRNA stability, and host gene expression are the main snoRNA abundance determinants in human, and it seems to be the case also for several other vertebrates. And we also saw that the more snoRNAs a genome contains, the smaller is the proportion of expressed snoRNAs in that species. So it means that a small subset of important snoRNAs needs to be conserved and expressed. The approximate 2/3 of the rest constitute potentially a pool to develop new functionalities as mutations accumulate, or either this snoRNA just become a snoRNA pseudogene. With that, I'd like to thank my lab and the funding sources and you for your attention.